So welcome everyone. Today we are going to see the program TY subject botany semester 5th, and the course code is BOC 1007, that is the microbiology and plant pathology. So myself Shubha Kautankar, assistant professor, Government College of Arts, Science and Commerce, Sanglim, Goa. The title that we are going to see today is Pathogen Attack and the Defense Mechanism, and the module name is Biochemical Defense Mechanism in Plants, that is pre-existing and induced. Outline of the lecture, that is introduction, then pre-existing chemical defense and the induced chemical defense. Learning outcomes will discusses the concept of biochemical defenses in plants, then it explains the role of pre-existing defense mechanisms, and it describes the functioning of induced defense mechanisms. Coming to the introduction, defense mechanism in plant also includes the chemical substances produced in the plant cell before or after the infection. So it has now been established that biochemical defense mechanisms play a more important role than the structural defense mechanisms. This has been supplemented by the fact that many pathogens entering non-host plants naturally or artificially inoculated fail to cause infection in absence of any structural barriers. Since they lack an immune system comparable to animals, they have developed a stunning array of structural, chemical and protein-based defense mechanisms designed to detect invading organisms and stop them before they are able to cause extensive damage. So the biochemical defense mechanisms are divided into two types, that is pre-existing biochemical defenses and the induced biochemical defenses. Pre-existing biochemical defenses are again classified into three types, that is preformed inhibitors, then phenolics and phytoalexins. Induced biochemical defenses are classified into two types, that is hypersensitive responses, or also known as hypersensitivity, and a systemic acquired resistance. Coming to the pre-existing biochemical defenses, the first one is the preformed inhibitors. So the preformed inhibitors are classified again into two types. First one is the inhibitors released in the pre-penetration stage. In this, the plant generally exudes organic substance through above ground parts, that is the phyllosphere, and the roots region, that is the rhizosphere. Some of the compounds released by some plants are known to have an inhibitory effect on certain pathogens during the pre-penetration stage. So for example, fungistatic chemicals released by tomato and sugar beet prevent the germination of botrytis, that is a kind of a fungus, and also a cercospora fungus. Presence of phenolics like protocatechoic acid and catechol in scales of red onion variety inhibits the germination of conidia of Colatotrichum cercinans and on the surface of the red onion. Presence of certain phenolics, tannins and some fatty acid like compounds such as dienes in cells of young fruits, leaves and seeds afford them resistance to botrytis. So several other compounds like saponin, tomatin in tomato and evanescin in oats have antifungal activity. So some hydrolytic enzymes like glucanases and chitinases prevent in cells of some plants may break down the cell wall component of pathogens. So these substances are stored in vacuoles, lysogenous glands and heartwood periderm of plants. Coming to the second part, that is the lack of nutrient essential for the pathogen. So it is another pre-existing biochemical defense mechanism. So plant species which do not produce any of the chemical essential for the growth of pathogen may act as a resistant variety. So for example, a substance present in seedling variety which is susceptible to the rhizoctonia, this is a kind of fungus that initiates the hyphae cushion formation from which the fungus sends penetration hyphae inside the host plant. So when this substance is not present, the hyphal cushions are not formed and the infection does not occur. The next part of the pre-existing biochemical defenses is the phenolics. So defense through increased level of phenolics is the another type wherein plant contains aromatic acids that act as a precursor of phenolic compounds. So phenolic synthesis takes place through shikimic acid pathway and the acetic acid pathway. These are the simplest compound that is which have the single OH group on benzene ring. So when benzene ring has more than one OH group, that is the polyphenols such as lignines, phenyl glucoside, flavonoids, anthocyanins, etc. So plant metabolites are the substances produced by all the plant cells that are directly involved in growth, development and reproduction. So for example, sugar, protein, amino acids, nucleic acids. Then the secondary metabolites, so these are not directly involved in the plant growth or reproduction, but they are involved in the plant defenses. So these compounds belong to three large classes, that is terpenoids, phenolics and alkaloids. So furanocoumarins are phenolic compounds which are produced by wide varieties of plants in response to pathogens or herbivore attack. So they are inactivated by UV light. Nitrogen compounds, so these are alkaloids and a large class of bitter tasting nitrogenous compounds that are found in many vascular plants including caffeine, cocaine, morphine and nicotine. So they are derived of amino acid aspartate, lysine, tyrosine and tryptone and shows effect on animal physiology. Some members of Solanaceae family produce many alkaloid compounds. So nicotine is an alkaloid that is produced in the roots of tobacco plant, that is the Nicotiana tabacum, and transported into leaves where it is stored in vacuoles. So it is released when herbivores graze on the leaves. Coming to the next, also caffeine is an alkaloid found in plants such as coffee, that is Coffea arabica, then tea, Camellia sinensis and cocoa, that is Theobroma cacao. Then these substances are toxic to certain insects and fungi. Then pyrethrin. So monoterpenoids ester produced by chrysanthemum plant that act as insect neurotoxin but are harmless to humans. Diterpenoids such as gossypol, a terpenoid produced by cotton, that is Gossypium hirsutum, that has strong antifungal and antibacterial properties. Monoterpenoids and sesquiterpenoid are the primary component of essential oils. So they are highly volatile compound that contribute to the essence, which often function as insect toxin and many protect against the fungal and bacterial attack. Then the last part of pre-existing biochemical defenses is the phytoalexins. So this concept was given by Boger and Muller in potato late blight interactions. So this is defined as the antibiotics which are produced in plant due to pathogen interaction or as a response to injury or physiological stimuli. So Paxton defined phytoalexins as low molecular weight antimicrobial compounds which are synthesized by and accumulates in the plant cell after microbial infection. This involves the role of elicitors in their production. Many phytoalexins have been isolated from following plant families such as Leguminosae, Solanaceae, Malvaceae, Poaceae and Asteraceae. Certain characteristics of phytoalexins. So the phytoalexin should be fungistatic and bacteriostatic and active at very low concentration. This should be produced by the host in response to infection or metabolic by product of microorganisms and stimuli. Also absent in healthy cells and present in very minute quantity. It usually remains very close to the site of their production and it produced relatively quickly in cells after infection. So it is host specific rather than pathogen specific. Then the best studied phytoalexins are, first one, they are classified into different types, that is legumes, Solanaceae and Orchidaceae. So in legumes, phaseolin and phaseolidin are found in common beans. Then vestitol is another phytoalexin which is found in the red clover, and picitin is found in the pea plant. Coming to the Solanaceae family, riscitin is found in potato and tomato, and capsidol is found in chilies. And in Orchidaceae, orchinol is another phytoalexin which is mostly found in orchids. Then the next type of biochemical defense is the induced biochemical defenses. These are classified into two types, that is hypersensitive responses, or also known as hypersensitivity, and the systemic acquired resistance. So coming to the hypersensitive response. Hypersensitive response is a mechanism which is used by the plants to prevent the spread of infection by microbial pathogens. It is characterized by the rapid death of host cells in the local regions at the site of infection, hence considered to be a type of programmed cell death. Hypersensitive response, or in short HR, is the result of a specific recognition of the pathogen attack by a host. It serves to restrict the growth and spread of pathogens to other parts of the plant. The hypersensitive response acts by isolation of the pathogen and it stops the flow of nutrients and thus leads to starvation. Certain characteristics of hypersensitive responses are, the hypersensitive response is the culmination of the plant defense which is initiated by the recognition of the plant of specific pathogen which is produced signal molecules which are known as the elicitors. So hypersensitivity has been studied chiefly in rust, then powdery mildews and potato blight, warts, etc. So in the incompatible host, parasite reaction, hypersensitive reaction takes place through quick necrosis of the infected plant. After infection in the resistant plants, browning or death of the infection of the cell occurs very rapidly. During this, loss of permeability of cell membrane, accumulation of oxidation of phenolic substances, then increased respiration, protoplasmic streaming, granulation, migration, swelling and disorganization of nuclei and appearance of brown pigment, etc. occurs. Then the next type of induced biochemical defenses is the systemic acquired resistance. So systemic acquired resistance is the activation of defenses in distal non-infected parts of the plant. So systemic acquired resistance confers broad-based resistance to the different pathogens. For example, primary inoculation with a fungal leaf spot pathogen reduces the susceptibility of the host plant to the other fungi as well as to the bacteria and viral pathogens. Salicylic acid is a part of signaling pathway which is involved in the transmission of the defense response throughout the plant to produce this systemic acquired resistance. Then coming to the induction of systemic acquired resistance, production of hydrogen peroxide has been associated secondary induction of systemic acquired resistance and direct toxic activity to invading pathogens. It also leads to thickening of the plant cell wall and accumulation of pathogenesis related to the proteins, that is the PR protein, chitinases and beta-1,3-glucanases. So this enzyme accumulate in vacuoles of plant cell. Upon attack, they directly degrade the fungal cell walls and directly their action results in the release of fungal cell wall component that elicit the additional defense reactions. Coming to the pathogenesis related proteins, or short the PR protein. So these are a structurally diverse group of plant protein that are toxic to invading fungal pathogens. They are widely distributed in plant in trace amount but are produced in greater concentration following pathogen attack or stress. So the pathogenesis related protein or the PR proteins are induced in a part of systemic acquired resistance. Infection activate genes that produce PR proteins. So some of the proteins are microbial, attacking molecules in the cell wall of the bacterium or fungus. Significance of PR protein. So they show strong antifungal and antimicrobial activity. Some of them inhibit spore release and germination. Others are associated with strengthening of the host cell wall and its outgrowth and papillae. So with this we have covered this chapter, and following are the references. This is the web link which I have referred, and thank you.